Going back to the control of hormones, what we've just been discussing, keep in mind, was the upstream release of hormones, and that's one side of the equation. But remember that there is a downstream reception of the hormones as well. And when we talk about the reception of hormones, we're going to be talking about target cells. So it turns out that hormones, remember that I said that hormones contact basically any cell in the body, especially any cell that has contact with vascularity, which pretty much all of our tissues do. And so since hormones are released ubiquitously through the bloodstream, they in turn will contact all cells of the body. But the only cells that will actually respond to the hormone are the ones that have the correct receptor for that particular hormone. And these cells would be called target cells. Okay, so here they are. We have our little hormone that's making its way through the bloodstream, and here's a nice capillary bed. So some of the hormone leaves the bloodstream at the capillary bed and basically makes its way through the interstitial fluid, all the fluids that are in the tissues, and they come into contact. You'll notice that this hormone has a little pointy, a pointy on it. That shape is important. That shape then fits perfectly inside of this receptor here, which is also, it's complementarily shaped to the pointy, whereas this guy over here, you'll notice this particular receptor is not shaped correctly to receive that particular hormone. So the pointy hormones will only fit with this particular receptor. Okay, so let me read the definition here. Target cells. Hormones may contact many different cells throughout the body, but they will only effect changes in certain ones that have receptors for the particular hormone. And so the receptors there's two different ways that you can have receptors. Receptors in this case are always going to be proteins or protein-based structures. And there are two places they can be. The one that we're looking at here is a membrane-bound receptor. We call it transmembrane receptor. It goes from one side of the membrane to the other. So this is an entire cell. This blue line here represents the cell membrane or the plasma membrane of the cell. And embedded in that plasma membrane, you have these receptors. Now, the beauty of these receptors that are membrane-bound is that they are able to receive signals from outside of the body. But just so you know, we're also going to be looking at some receptors that actually live inside of the cytoplasm or possibly right directly inside of the nucleus of the cell. And it's still a protein. It's still a protein-based structure. It still has a particular shape to it that receives receptors, but they work a little bit differently from these guys that we're looking at here. So I want to talk for a moment about specificity. There's sometimes some hormones will only have a very specific level of number of target cells. So any cell that has the receptors for a particular hormone are called target cells. Those cells that do not have those receptors are called non-target cells. And so what I wanted to say was that some hormones have a very large number of target cells and some have very few. And so that's what I'm referring to when I talk about specificity. So for example, there's a hormone that is called ACTH, which is adrenocorticotropic hormone. That hormone has a very small number of target cells. ACTH only targets particular cells that are in the adrenal cortex. Whereas a hormone such as the thyroid hormone, which is our primary metabolic hormone, basically affects any cell of the body. So the specificity there is quite broad. It actually has almost no specificity at all. Thyroid hormone causes changes to occur in almost every cell of the body. Let's talk about upregulation and downregulation of these receptors. So keeping in mind, we're starting to use this nomenclature a lot. Target cells are the ones that actually can receive the hormone. And remember, what makes a cell a target cell is that it has the particular receptors for that particular hormone. So let's talk about upregulation. The cells have an ability to become more sensitive to a particular hormone, or they have the ability to become less sensitive to a particular hormone. And the way that they would do this is simply manufacture more receptors. Remember that I said receptors are protein-based. Remember where proteins come from. DNA transcribed into RNA, RNA translated into proteins, and there it is. So all of these receptors have a particular gene that is encoded in the DNA that rests inside of the nucleus, and so we can just transcribe more of them or upregulate them when we want to receive more signal or become more sensitive to that signal. So in this case, you have a target cell that only has 2 receptors shown. And what you can do is you can upregulate the receptors to 4 times as many in this case, which would be 8 of them in our little picture. And if you have 8 receptors versus 2, this cell has now become more sensitive to that particular hormone. And that can change over time. It can change over different periods of time in a person's life, or it can change with different needs that the body has. It just depends on what's going on. The same thing can happen here. We have downregulation. Here we have a target cell which is quite sensitive to this particular hormone, and over time some of those proteins began degrading, which is a natural process. And in this case, this particular cell did not manufacture any new ones, and so this cell is becoming less and less sensitive to that particular hormone as time went on. So look over the list of hormones. We'll be discussing lots of different hormones in class and in lab, and we'll be thinking about what they do, what the action of those hormones are, kind of putting a practical application into what the hormones are. I want you to go back to this idea of upregulation and downregulation. Think about particular hormones. Why would it be that a particular hormone might want to have a more profound effect on its target cells at certain times and less of a profound effect at other times? I want you to think of examples in your own head and then bring those examples to class.